no iGEM team has done this before.
With our contact lens and our device, we are able to save these people from blindness. Hello and welcome to Einstein News. It's a beautiful day, but we might not see this lovely world any longer. I'm Yiling with the latest headline. The criminal who identifies himself as Mr. G is still at large. He's the head of notorious Syndicate X, where he and his members have been charged with the assault of over 18 million people by causing damage to their vision. His mode of operation involves affecting victims with a disease called glaucoma. Glaucoma is an ocular disease that causes irreversible damage to the optic nerve. Without early intervention, the victims can only end up blind, causing glaucoma to be the leading cause of irreversible blindness. What's even worse, since there are no early symptoms of glaucoma, no one knows of its presence, which earned glaucoma the moniker the silent killer of vision. According to our sources, the US government alone pays almost $3 billion every year for glaucoma treatment, and there are 18 million people globally affected by glaucoma. Now, let's take a look at how you can get glaucoma. One of the major risk factors is the elevation of intraocular pressure, IOP, which is caused by a buildup of aqueous humor inside the eye. Aqueous humor is the circulating fluid within the anterior chamber of the eye, which is secreted by the ciliary body and drains through the trabecular meshwork. However, if there's an imbalance between the rate of secretion and drainage, the aqueous humor will build up and increase IOP. The increase of IOP will damage the optic nerve, leading to blindness that cannot be reversed. Now, let's move on to some questions that the public may have.
Good afternoon, Einstein News. This is Mr. G speaking. It has come to my attention that you're doing a segment on my amazing achievement. Word of warning, however, move too close and you'll find your vision gone forever. Keep an eye out for me.
What just happened? Sorry, what? Okay, I see. We have just received news that a team from Taiwan has managed to find a solution for this disease. It seems like we have no need to fear Mr. G and Syndicate X any longer. Our regional reporter, Felicia, reporting live from NCKU Tainan HQ.
This is Felicia, reporting live from NCKU Tainan headquarters. We have received news that this undergraduate team has found a way to treat glaucoma. Now, let's head inside and find out how they managed to come up with the solution. Here, with us here is Haley. Could you introduce yourself a bit?
Sure. Hi, I'm Haley from iGEM NCKU Tainan 2020, an undergraduate team who managed to develop a groundbreaking treatment for glaucoma. I know to slow down glaucoma progression. I know is a pair of contact lenses that contains engineered E. coli, capable of releasing drugs to lower the intraocular pressure, or IOP. The lens is fitted with a ring-like compartment made of semi-permeable membrane that is filled with our engineered E. coli, WN3064, L-arginine, IPTG, arabinose, and diaminopermalate, DAP.
Wow, that's an interesting approach. But why do we need to lower IOP to treat glaucoma?
Well, inside the cornea is a circulating fluid called aqueous humor. If it accumulates, this fluid will cause pressure on our optic nerves. Since we know that there is a smooth muscle layer called the trabecular meshwork that controls aqueous humor outflow, we designed a treatment that targets it. All we need to do is relax that muscle and let aqueous humor flow out. I know it's able to achieve that by releasing nitric oxide, a non-muscle relaxant.
Oh, so how do you produce nitric oxide inside the contact lens?
We specifically engineered our E. coli to do so by cloning the nitric oxide synthesis LS gene from Bacillus subtilis, which is an enzyme that converts L-arginine into nitric oxide. As you can see in this SDS page, we successfully express LS in our engineered E. coli. Since our LP fluctuates constantly throughout the day, we want to ensure that the nitric oxide is produced only when LP spikes. To do that, we introduce two plasmids into the bacteria. The first plasmid contains T7 RNA polymerase to regulate the gene expression of the second plasmid, which contains an IPTG-inducible system to control the production of LS. When IOP increases, the contact lens will deform, thus pushing out water from the ringlet compartment. This will result in an increase of IPTG concentration, which then induces more expression of LS.
So iKnow works like a dynamic drug delivery system. While the principle itself seems to work, how do you prove your design works?
Yes, we demonstrated our design works in three parts: lens deformation, IPTG induction, and nitric oxide diffusion. Now, let me show you. We first set out an experiment using the pulsing eye to model the relationship between IOP and IPTG concentration change in our lens, based on Pascal's principle of fluid mechanics. Changing the height of the drip bag will increase the water flow to the eye, thus controlling the IOP. Combined with DIC, a digital image correlation system, we can know how much it has deformed. The data shows when there is a 10 mm of mercury elevation in IOP, the deformation will be up to 2.9%. That is a 3% difference in IPTG concentration. We conducted an experiment using an LS assay kit. The result shows that the increase of IPTG concentration will lead to higher LS expression and also more nitric oxide. Finally, we placed contact lens onto the pulsing eye, dripped LS and X substrate onto the contact lens. After 1.5 hours of incubation, we extracted the aqueous humor and measured nitric oxide concentration. As seen in this result, we proved that the nitric oxide can diffuse into the aqueous humor successfully. We also set out a diffusion model to predict the time needed for iKnow to reach the minimum effective concentration of nitric oxide and lower intraocular pressure effectively. We divided the nitric oxide diffusion pathway into 3 layers: the contact lens, the cornea, and the aqueous humor. The result shows that it only takes about 30 seconds for the nitric oxide to reach a concentration that is effective for treatment. Packing these proven designs into our innovative contact lens, we have a product that can relax the trabecular meshwork when IOP is high. Next, my teammate Virginia will share more about the design of iKnow.
Okay, we want to ensure our bacteria can be stored in the contact lens until it is ready to be used. We incorporated a Hig-A Hig-B toxin antitoxin system and recombinase into our growth switch system. This design enables us to control bacterial growth in 3 stages: production, storage, and medication. During the production stage, the bacteria will grow normally. Then, for storage, we will induce the Hig-A toxin to cause the bacteria to hibernate. Finally, when it will be used for medication, the bacteria will be activated. The body heat will induce the recombinase system to delete the Hig-A toxin gene and causing the bacteria to resuscitate and start producing the therapeutic agent.
That's amazing! And I'm sure you have considered iKnow's biosafety. Can you tell us more?
Yes, of course. To avoid the risk of bacteria leaving a damaged contact lens, we utilize a curly proteins that can form a fiber-like structures on the bacteria surface. Inspired by previous iGEM team who expressed CSGD, a transcription factor of curly proteins, we improve it by changing the promoter into PLEC and adding CSGA to act as an anchor for bacterial attachment. By producing more fiber-like structures and anchoring them to the contact lens, we are able to increase our bacteria's grip on the contact lens. In our experiment, we have measured our engineered bacteria's binding affinity. We first define binding ability as a bacteria's resistance to external forces. Therefore, after culturing the bacteria in a 96-well plate, we invert the plate and give an external force by dropping a book just like this. Then, we resuspended the bacteria and measured its OD600 value to determine its binding affinity. Through this graph, we have proved that co-expressing CSGA and CSGD has the best bacterial attachment.
So CSGA-CSGD co-expression will ensure the bacteria to stay attached on the contact lens. But what if the bacteria actually escape to the atmosphere?
Oh, it's nothing to worry about. We have taken another precaution to ensure the safety of INO by using WM3064 strand as our chassis, because the DAP agent has been knocked out. The DAP agent encodes for an essential enzyme necessary for cell wall synthesis and growth. This will cause the bacteria to have to depend on exogenous DAP in order to survive. Therefore, we added DAP inside the contact lens to ensure its survival. And if it leaves the contact lens, since there's no DAP in the environment, the bacteria won't survive. As you can see in this plate, the bacteria only grow on plates with DAP. So, from inducing nitric oxide production, constructing growth switch system, to ensuring its biosafety, we have provided remarkable results to prove the success of INO.
Thank you for the explanation of INO. It's really innovative. And I heard your team also designed a device.
Why? Yes, my teammate Albert will explain about the device.
Hi, I'm Albert. As mentioned before, glaucoma causes irreversible damage to optic nerve. Since there are no early symptoms of glaucoma, the public is unaware of its presence. Therefore, early detection is needed for glaucoma patients. Measuring IOP is the most common method to predict glaucoma. The current IOP measurement techniques exert pressure on the cornea, causing discomfort to patients. To promote glaucoma detections and verify INO's effectiveness, we created iScreen.
Do you mean this one?
Of course not. iScreen is a portable and low-cost device providing pinless IOP measurement. It only costs about $55.
I see. So how does iScreen measure the IOP?
It can transmit ultrasonic waves into the cornea, analyze the reflected signal, and generate IOP reading in real time. Here's how it works. First, our custom function generator will drive 40 kHz transducer to transmit ultrasonic wave. With the amplifier and the envelope detector modify the reflected signal. Then, we convert the signal into the corresponding IOP value with Arduino. Finally, the IOP and risk level will be shown on the LCD.
Cool. So how do users operate it?
iScreen is extremely user-friendly. Just hold it in front of your eyes. Push the button and wait for a few seconds until you hear a beep. Also, we recommend our user to download iCloud. It's an app we developed to record users' IOP and upload it to their personal ThinkSpeak account. First, download the app before using iScreen. Touch the Start, and it will ask you if you have applied for a ThinkSpeak account. Enter the URL. Your data will automatically upload to ThinkSpeak. Touch Bluetooth. Connect to iScreen. Now we are ready to start. Push the Start button again. Wait for a few seconds. The result will be shown on the screen. The whole system is convenient for long-term tracking and observation. In addition, it can be used as a tool for large-scale data collection to assist the development of INO or other research including IOP.
Thank you, Albert. With iScreen, I believe NCKU Tainan can raise the public willingness to measure their IOP, preventing more people suffering from glaucoma. And finally, Mr. G won't be tried anymore. Thank you for the intriguing introduction from NCKU Tainan. It's really amazing what they have accomplished. However, while developing iKnow and iScreen, NCKU Tainan realized they had a much more challenging problem to solve. Most people simply do not know of glaucoma. In order to benefit the whole of society, they came up with different programs and activities to raise the awareness of glaucoma as well as synthetic biology. They aim to educate people of different ages, children, adolescents, and elderly. Therefore, by adjusting the way of teaching them according to their age and knowledge, NCKU Tainan showed that everyone could understand synthetic biology and glaucoma. Additionally, by utilizing social media, they can reach out to even more people. First, during the Glaucoma Awareness Week, they shared about glaucoma facts in social media. Second, they also created the Something podcast, introducing the things that revolve around iGEM and how synthetic biology can help the world. Lastly, they introduced synthetic biology in 5 levels, a video where they explain synthetic biology to people with 5 different levels of understanding. Students from middle school, high school, college, grad school, and non-experts. Furthermore, to raise the awareness of blindness, NCKU Tainan made a LARP or live-action role-playing event called Into the Darkness. With an intriguing storyline and sensory engaging experience, Into the Darkness was a massive hit. Not only were players able to experience the life of the visually impaired, they were also able to learn about synthetic biology in a new and fresh approach. Moreover, they collaborated with many teams who have similar ambitions to solve local and global problems. In particular, they collaborated with CSMU Taiwan to launch I've Got a PhD, a social media platform sharing topics related to the 3rd and 4th SDGs, which are good health and well-being and quality education. Through this platform, they were able to work with more teams to convey correct information for the public. Also, the 2 teams worked together to hold Taiwan iGEM panel discussion about SDGs to encourage college students to contribute to these goals. Finally, to realize their design, they contacted the National Applied Research Laboratories in Taiwan and visited Taiwan's leading contact lenses manufacturer, Chemex Optical Corporation, to understand the structure and production of the contact lens. iKnow and iScreen is more than just a concept. Not only is NCKU Tainan envisioning the future for glaucoma treatment, they are working hard to make it a reality. From educations by lectures, sharing through social media platform, special lab event, to collaborations and consulting with the experts, NCKU Tainan alone has done many marvelous things to create an impact on society. Can you imagine what will happen if there are more teams and people are doing the same? A revolution is coming soon, with NCKU Tainan aiming to lead the charge. I cannot wait to see what NCKU Tainan and the rest of the world will do next. This is Felicia, reporting live from NCKU Tainan headquarters. Now, back to you, Yiling.
Thank you, Felicia. Well, there you have it, folks. Thanks to NCKU Tainan's iKnow and iScreen, we no longer need to worry about Mr. G and Syndicate X's nefarious plans to bring darkness to the world. And Mr. G, better keep an eye out. I think your time is running out. This has been Einstein News with Yiling. Thank you for staying with us. Bye.